Okay. We're talking about, we've reached that part of neural development where there's plenty of neurons, plenty of targets that sent out their axons. Their axons found their way to the more or less correct target. We'll see later that fine-tuning is going to go on basically the rest of your life. But now the initial synapses that have been made, they need to mature well enough to actually function properly. And part of that process is to get rid of connections that are inappropriate or aren't useful. And we'll come back to that topic when we get to the old chapter 8 and new chapter 9. That this continuous loss of synapses that might seem regressive to you, but in fact is an important part of maturation. We have more synapses than we need earlier in our life. And to the extent that we develop more and more sophisticated behavior, it's because we're shedding connections that were not helpful. And we were talking about Fragile X syndrome, which is caused by problem on a gene on the X chromosome, a case of trinucleotide repeats that tend to get larger over time, or can, and if they're too large, the gene doesn't work properly. And in the case of Fragile X, the gene is called Fragile X mental retardation 1, because it was the first one they found, and it turned out to be pretty clear that it was the one that was indeed causing the syndrome. So the syndrome, I haven't talked about it a lot. Fragile X syndrome is one in which there's a certain effect on the face, facial development. People with Fragile X tend to have kind of long faces, large ears, things like that. Another case of pleiotropy, right? Who would think that a gene that's absolutely crucial for getting rid of bad synapses also plays a role in how one's face gets put together, but that's totally commonplace in development and in genetics. And it is usually accompanied by some degree of intellectual disability, although that can vary widely. And as we say here, they often have extremely flexible joint. So there's another pleiotropic aspect of this. And they also tend to be somewhat clumsy, so ataxia, and they might have low muscle tone. The muscles don't tone up very much, right? So they tend to have sort of flabby physique. So wide-ranging influences affect, and almost certainly the effects that cause intellectual disability are those that are happening in the brain. And the one thing that we can find that's different about people with Fragile X versus controls is they have more synapses in the cortex. If we look at, an interesting thing happens because of our old friend regulation, that when there's too many repeats and the transcripts aren't working very well, the brains tend to produce more. So there's more and more of these transcripts trying to get enough functional protein out there to do the job. But, and so if we look at a graph here that was put together, if we have here how many repeats there are total, and if there's less than 55 repeats, there's basically no symptom, nothing that you can see. But once you start getting more repeats, this here, the red, we're keeping track of how much mRNA is being made. And this is what I meant was, as the repeats get bigger, there's more and more transcripts getting made. But the problem is, it's not doing its work. And after you reach a certain point, even the compensation for mRNAs, too many repeats, and even that compensation tends to go away. And this is the point at which you really start to see lots of behavioral cognitive deficits. When you know how many repeats a person has beforehand, people can show that you can find some evidence of subtle symptoms there that would probably go undetected if they weren't being looked for really closely. Fragile X is much more common in males than it is in females. because males only have one X chromosome and females have two. So it's very unusual for two X chromosomes with lots of repeats on each to end up in the same zygote and produce a girl. But if mom has a lot of repeats on one of her X chromosomes, and if that's the X chromosome she gives to her son, then he will have this syndrome. And yeah. Questions about that? Oh, and here's just another. I mean, it's pretty, yeah, Justin.
It was my understanding that the CGG islands cause this disorder by like doing or ubiquitin binding, preventing translation. So how come if there's more CGG, it's able to create more of the mRNA? Does it have something to do with stability?
Are you talking about why the number of mRNAs goes down afterwards, or
why it goes up?
Because whatever's happening to the transcripts that are getting made, the cells have some way of noting it, I don't know how, and start calling for more transcription.
Okay, so more like proteins to transcribe it. Those CGGs aren't actually translated then?
I don't remember if they are or not. No, I think they are, and the protein can handle a certain number of them, but not past a certain point. But I'm not really sure where they get translated.
So it could be mRNA stability, or it could be like whether it brings transcription factors over and
right. And I don't know. If I knew, I forgot. Yeah, Allison.
It's the fragile syndrome directly related to the human growth.
Is it directly related to?
And then Earth.
Yes. I mean, not directly. It's not coding for that. It's, in fact, what it's holding on to is it's usually the protein inhibits certain things that are important for development. And so probably what it's doing is it's keeping things in scarce supply. And when it's not functioning properly, there's more supply, less competition for synapses, more synapses stay. And that's probably what's causing the intellectual disability. Maria, did we get to your?
Well, are the repeats like inserted, or are those?
Yeah. At what stage do the repeats? Because sometimes there's so-called progression where the parent will have a certain number of repeats, but some of their zygotes will have more. And again, I don't remember at what stage that happens, whether it's the reduction of the zygote or, I mean, it has to be. And where in the production of the zygote the repeat happens, I don't know. Yeah. And so people have done these sorts of mutations and inserted them in mice. And when you do that, indeed, you see a lot more synapses in the cortex. And the mice, also have behavioral deficits. They don't learn as well. So it seems pretty clear that that's a connection. You can't actually prove that it's additional synapses that causes the behavioral deficit, but the correlation seems pretty good. And there. Okay. Ready for a Kahoot then? Hope to get 2 in today. We'll see. So some of you probably read that the Nobel Prize in Physiology and Medicine was announced a day or two ago. What was the nature of the discovery? That, yeah. Oh. About mRNA, yeah. About the existence and importance of smaller RNAs, and the significance of it is this is a way of controlling transcription. So when I tell you in development, gene expression is the end all and be all, it still is, right? And that's still cutting edge research is what's controlling gene expression. I don't know. If they didn't even offer me even a share of the prize, so I don't remember it. And I've never done any work with RNA. Okay, everybody in that wants to be in. Well, hey. Yeah, it's catenin. That's an intracellular protein, right? So it can't be in the extracellular matrix. It's not secreted.
Zero.
So Allison, did I get to your, well, I'll let you answer that next. And answer your question about, so is the Fragile X, is it directly inhibiting transcripts for catenin? Not that I know of, but it is inhibiting other requirements. Oh, okay. So I wasn't even, scaffolding is the term being used, right? So, and especially important in the postsynaptic cell. They do anchor, true, but the terminology used is scaffolding.
Yes, you break over. Be trying.
Yeah. So first contact, then start assembling things, and then stabilize the synapse. And as we're going to see next, even after the synapse is stabilized during development, there's going to be big changes. You can't change? Oh? Well, you've got enough candy. Yeah, duplication and divergence. You can't get much divergence unless there's a duplicate yet, because the divergent will make that gene dysfunctional and will get selected out, right? But if there's a duplication, then one can start changing, and you still have function because of the other copy. So one tends to stay still, and the other may accumulate more and more mutations, more and more changes in function.
Thanks.
Yep. Beta-catenin is one of the things we know they're competing for. And from the Fragile X, there's also reason to think they're competing for other proteins, including scaffolding protein. So I could have substituted scaffolding proteins instead of beta-catenin in there. And hopefully that's not the next question. What is it? Step 2 answers. Yeah, twisted synapses. Where did that come from? Justin, you read the book beforehand. I don't remember talking about. I don't want it to You know what? Just try to keep people on your toe. So hopefully I didn't cheat someone out of a Zettelman. What's your name? Evelyn? Okay, good. Who's number 2? I know. Great. Oh, almost got to you. Who's number 1? Allison? Okay, see, I answered your question just in time. Okay. Oh, so great to lose that soundtrack. Okay. So now we're going to go on and talk about the individual components that are known to be involved in synapse stabilization and growth. And first, let's take a look at some of the proteins at your everyday synapse. Okay. So we mentioned before, initial contact is usually being mediated by adhesive factors. So there's some match between the adhesive factor that's being produced, being inserted in the membrane of the presynaptic cell, and that being produced by the postsynaptic cell. Often it's homophilic affiliation, and therefore it could be the same. And among those things we've talked about the cadherins and protocadherins, and I don't think we'll talk about many more. I guess, yeah, L1, NCAM, things like that. So, and this is the picture we've seen before about how these things are attached. And there's not much in the way of presynaptic machinery, and initially at least, there might be no receptors whatsoever. But then there'll be inductive factors that are happening. And so there are 2 crucial proteins for a synapse. There are, and as usual, in invertebrates, there's only one of each, but in vertebrates, there's a whole family of each of them. So there are the neurexins, which are getting made by the neurexins that are getting made by the presynaptic, and the neuroligands that are being made by the postsynaptic cell. And they like to stick to each other. If you want a mnemonic for how to remember which is which, one I use is neurexin is being made by the end that's making exocytosis, right? Synapse neurotransmitter release. And neuroligand is being made by the cell that's going to bind ligands at the synapse. And they start binding to each other. And once that happens, in the postsynaptic cell, there's the development of a postsynaptic density with all sorts of proteins being assembled there, making a scaffold, the scaffolding proteins, the postsynaptic density protein, and there's a family of them, and they can tend to be named after how big they are. So PSD 95 is 95 kilodaltons in size, etc. I don't need you to remember any of that. I just want you to know about PSDs and the like. And what happens on the postsynaptic side is all these proteins are getting glommed together, holding on to each other to keep the whole unit together. And we're not showing it here, but at some point, this is also being connected, often through beta-catenin, to actin that is itself part of the cytoskeleton of the cell. So that means once the synapse gets made, it doesn't float off somewhere else. because remember, all these proteins are basically dissolved. They're proteins in which one part of the protein is dissolved in the lipid bilayer because it's especially lipophilic, as you've run into before. After that happens, then the postsynaptic cell will start putting together various receptors to insert into the synapse. Here we're showing two glutamate receptors, the so-called NMDA receptor and the AMPA receptor. And there's this picture we're showing here of all these proteins holding on to each other is a gross simplification. It's really a total gemisch. Lots of different players, lots of different sticking, lots of different cross-linking, all of which is going to keep the thing together. And what's interesting about that is later we're going to see that synapses, sometimes in humans say, synapses that have been present for years will go away again, which means all that work has to get disassembled again. Not our topic for today. And then as that happens, now the presynaptic cell, again in response to the binding of its norexin receptors, starts assembling more and more synaptic machinery, more vesicles, more vesicles and things like that. Then, as one of the things that has to happen for this thing to work is, I'm not showing you that yet. Okay, questions about that. And this is also, just a reminder that there are microtubules in here that are also part of the cytoskeleton, holding things together, holding the shape of the cell, and keeping the synapse where it needs to be. What else do I need? And eventually there have to be calcium channels in the presynaptic cell because it'll be the influx of calcium that will trigger synaptic release. So, in the early days of looking for neuroligand and norexin, people were finding more and more proteins that were associated with synapses, and there was an attempt to figure out which ones are important and which ones are just along for the ride. And so in one experiment that we talk about in the book, they showed the importance of neuroligand by transfecting non-neural cells with norexin. And when they did and put them in vitro with neurons, there would be postsynaptic densities appear on the cultured neuron wherever these cells that are expressing norexin happen to touch. And you didn't even need cells that were making norexin. You could coat beads, inert beads with norexin, or with neuroligand, and put them together. And then the question was, will cultured neurons, start placing norexin wherever a bead is hitting them that expresses neuroligand? And do anything start happening in the neurons as a result of it? And so in these experiments, what they found was that if the bead was coated with neuroligand, then the cell cultures, the neurons in culture that were touching them, would start making lots of synapsin in this case. And if the bead was just phosphate coated or lipid coated or just the bead alone, none of the others were able to increase how much of the synapsin the cells were making, right? So this one here is this was the baseline level of how much synapsin they saw. And you can see there's a 5-fold increase in that. And so that was how they showed that, in fact, neuroligand is the trigger to tell the, is the trigger that tells the presynaptic cell to start making the stuff you need to make a synapse. And that then gets us to so that experiment showed that neuroligand was sufficient to cause a synapse to start developing. And then they wanted to know if it was really necessary, right? Because again, maybe it's showing up, but it's not what's actually required. And then working in mammals, that became difficult to show because there were 3 different genes involved, 3 different neuroligand genes. And again, it seems to be an instance of redundancy, because when they deleted one of the genes, they saw almost no phenotype whatsoever. Nothing. And this was in the battle. Anyway, they knock out each of the 3 genes in turn, and you knock out only one, and the mice seem fine. Who cares? In those days, this was before CRISPR. In those days, the only way you could get a triple knockout, get a disabled gene, 3 different disabled genes in one animal, was through breeding. So you'd have to have all these breeding programs where you have this colony with gene A knocked out. I think they're actually numbered. Gene 1 numbered, knocked out. This colony, gene 2 knocked out. This colony, and keep breeding them back and forth and keep track of them until finally you get some animals where both copies of all 3 genes are defective. And those animals died at birth. You can't live without that, right? And probably the problem was that they couldn't breathe and stuff like that. But what they did show was that in those animals, if they took them out before birth and started studying brain connections, the synaptic transmission was almost 0. They could find almost no evidence of synaptic transmission, where they would stimulate some neurons that should be making a synapse with some others and see almost no response in the postsynaptic cells. How's that? And I'm not going to review it, but similar experiments were done for Rapsyn. This should be a review for you of the things that have to happen to have a well-functioning synapse. You've probably run into this before, but I do want you to know the names of some of these proteins. Unfortunately, they go by different names in different places in different literature, et cetera. So I'm trying to find, trying to go with something of a consensus. So they're the SNAREs, which are basically, you can just think of them as proteins that are important for synapse to develop. This is what they happen to mean: soluble in, now I'm not going to pronounce that, sensitive attachment receptors. And their job is just to latch on to various parts of the machinery that's got to work. So there are the V snares that will hold on to vesicles. So right here, for example, where do we have it? Come on, where's synaptic ribbon? Well, what? Oh, there it is. Thank you. Yeah, I shouldn't have had to put the label there. Anyway, the synaptic ribbon is an example, not the only one, of a V snare. So it's a snare that holds on to the vesicle. There'll be another snare that holds on to the postsynaptic membrane, and they have to bind to each other for the vesicle to get released eventually. So there are then, bless you, T snares that are on the target, meaning the postsynaptic membrane. And you've heard these names: syntaxin, SNAP25, things like that. And so if we take a look here, here's a syntaxin holding on to the postsynaptic membrane, synaptic brevin holding on to the vesicle, and they like to bind each other. And there's things like SNAP25 that will help them to bind together. And there is also attached to the vesicle, there's another protein you might have heard of called synaptotagmin. And the synaptotagmin basically serves as a calcium sensor. It is attached to the vesicle, and when calcium enters, it triggers the pulling together of the vesicle to the postsynaptic membrane, pulling them together enough so that the membranes merge, these 2 lipid bilayers, right? Just fat molecules. They shove each other aside, and eventually an opening gets made, and then all that neurotransmitter that's inside gets out. Yeah.
So the fundamental is communication between 2 cells.
Yes?
And the storage email, the inspiration already knows that it's regenerating, so it releases calcium.
No, I'm sorry. So I should have said that the calcium is going to come from, because an action potential is going to reach the bottom of the presynaptic. And so now, those calcium channels open, calcium enters, triggers this change, and the vesicle gets released. Allison?
Just to double check, so that's inductive factors like neuropsin and neuroligand, those are like the ones that stay, and the adhesive factors are the ones that are just like...
They certainly stay. The neuroligand and neurexin stay, but then what'd you ask? The what factors?
Adhesive factors.
Adhesive factors. Oh, well, no, the adhesive factors may often be there too, right? But we're not showing this in the cartoon because it's already confusing enough.
Do they both come at the same time?
No, the adhesive factors will be first.
Okay.
Absolutely. Because the synapse doesn't start assembling until some neurexin finds some neuroligand. Or vice versa. Great question. Lets me know that I'm going too fast. So, other questions about... And yeah, all the proteins I name here, I want you to remember what they are, right? And, synaptic tegments, the calcium sensor, etc. Yeah, I guess syntaxin for a T snare and synaptobrevin for a B snare. And yeah, there are more, but, I think I have a table of them here, but I don't want you to remember anything on those tables other than what we're saying here. Okay, something else. Oh, yeah. Botulinum, right? Botulism, botulinum problems. You open a can of soup and it's been contaminated and you failed to boil it entirely and you ate it anyway and you end up taking in some botulinum bacteria and they start making botulinum toxin and pretty soon you die because your breathing stops. Because the toxin chews up the synaptic machinery but vesicular release, and therefore the diaphragm muscles don't get the message anymore and you die. If you get found soon enough, they can rush you to the hospital, provide you on a ventilator, and let that breathe for you until the system clears this stuff out, and then you get to recover. So and specifically, it's chewing up all these proteins, pretty non-specifically. And without these proteins, the B snares, T snares, synaptic tegment, etc., you can't get vesicular release from the motor neurons onto your diaphragm muscles, and therefore breathing stops. So, if you're going to eat food from a can, make sure you boil it entirely, right?
Motion stopping.
Pardon?
Motion. But it's got not the ADG.
Yeah. I mean, you're also paralyzed, but you can survive being paralyzed a while as long as you keep breathing. Yeah.
Does it not pass into the brain then?
I don't know. I mean,
wouldn't that kill you? Like, wouldn't there be a lot of detrimental effects it takes?
Yeah. And as far as I know, I don't know what the mental symptoms are of people with botulinum toxin. They tend to be unconscious. So that's probably not helping things. But is that a secondary consequence of the fact that oxygen levels gotten low? I don't know. Yeah.
Or keep beating?
Yeah, apparently so. Yeah, Allison? And so you wonder, what's special about those synapses? Well, you can imagine they're special because, if they're not working, the game's over. But I don't really know the details. Allison?
Not eating ZKP, you should eat canned tuna because if you always eat that up with that.
Well, I mean, okay. So yeah, yes, you can, and people do all the time. And I don't know why it is. It's soup in particular. I mean, old people will, open a can of Campbell's. Surely they won't sue me for saying that. And they don't bring it to a complete boil because they don't want to be that hot anyway. And if it's a botulinum, then they end up dead. Yeah, Maria?
Isn't that's why I do hot.
Pardon?
Isn't botulinum toxin just Botox?
Oh, yes. And the protein, Botox, that's a good point. It's useful at, killing muscles and making them swell up and tissue. So the whole point of botulinum toxin is it inactivates the muscles for a while until the system recovers and causes inflammation, and it makes your wrinkles go away.
like it's like, yeah, it's harmful, but it's also like, I mean, it's not, it's like, not that it's not that harmful, but like, I know, for example, a lot of people who have a lot of like tension.
Uh-huh.
Well,
Yeah, Botox is a great way to inactivate a muscle for a while in various contexts. When it's done cosmetically, it's mainly to cause the skin to swell so that the wrinkles go away. Yeah.
how common is it?
How common is Botox?
No, like, it's
Botulinum? I don't know. I mean, Google it. I mean, people die of it in the United States every year. Is that
like, right now,
pardon?
like, it's
Let's see. Why is it safety tuna? I imagine because it's been packaged to make sure there'll be no botulinum in it, right? But I'm telling you, it's not safety soup if you don't. Yeah. If the directions say bring to a boil, I advise you to bring it to a boil. I didn't really mean to do a public service announcement about cooking soup, but you see how discursive my head works. So there you are. So what happens then is there's going to be a more and more buildup of the postsynaptic side, so-called postsynaptic density. There are some of the proteins that are involved. It's huge, right? 400 different proteins. It's been happening a long time. One example that you might run into once in a while are the membrane-associated guanylate kinases, and they bind to the intracellular neuroligand, right? That's the one on the postsynaptic side, and they bind to neurotransmitter receptors, and so they kind of keep them close together. So they're a way of lashing together the intracellular portion of the neurotransmitter receptors so they stay in place, stay there at the synapse. And later we'll find that muscles have their own special protein for lashing together acetylcholine receptors so that they stay at the neuromuscular junction. Other questions about that? So a whole bunch of molecular machinery has to come together for the synapse to start functioning as a synapse and not just a tentative touch between two cells. And so I'm going to switch topic a little bit here, unless more questions about that. Okay. We'll see repeatedly in this course, if you don't know this yet, in the history of neuroscience, most of what we have learned about synapses happened at neuromuscular junctions, the place where motor neuron axons touch a muscle and cause it to contract. And the reason that's where all the progress has taken place is because that's where the light is, for several reasons. Number one, the synapse isn't buried inside some cranium. It's sitting out there in the periphery. So you can do pretty simple surgery and open up of the skin, and there's a muscle and there's a line of synapses on it. Number two, neuromuscular junctions are huge. They're big enough that if you make them black and look at them on a muscle, you'll be able to see them with your naked eye. And they're also, they're one of the first neurotransmitters that was discovered, right? Otto Lowy and his fabulous dream where he discovers acetylcholine, which he called Vegas stuff. That was in the heart, but then it later turned out, in vertebrates at least, that's the neurotransmitter being used at neuromuscular junctions. And so we've known the most about acetylcholine and any other neurotransmitter. Another thing that's great about neuromuscular junctions is it's dead easy. With 19th century technology, you can detect the response to the muscle. Number one, you might just detect that it contracts. Or number two, you put yourself a little bit in the 20th century and you can stick a microelectrode inside a muscle fiber and you can record the postsynaptic response. And that's because as neural targets go, muscle fibers are huge. I keep using that word. They're so big, again, you can see them with the naked eye, and it's pretty easy to stick a microelectrode into one. I say all this to say that in science, often the most important variable to decide how to make progress is to find the right system that is tractable, in which you can actually figure out what's going on. And another philosophy of science that historians of science have noticed is that you want to find the most difficult problem you can find that is still tractable, for which you can make some progress. So, so over and over again, this is what the neuromuscular junction has done for neuroscience. To talk about the development of neuromuscular junctions, then we have to have a little aside about the development of muscles. One of the reasons why muscle fibers are huge is they are multi-nucleated cells. Mesoderm eventually starts making things called satellite cells, and muscles will retain some satellite cells all their lives. But some satellite cells, in response to some inductive factor, I don't know the details, although I think it's pretty well known, they start differentiating into myoblasts. So they take on a very characteristic look and a very characteristic shape, and myoblasts love to fuse with one another. And that's another thing that makes them special. And once they fuse with one another, they form what are known as myotubes. There's no contractile machinery yet. Actin and myosin hasn't put together yet, but it's a huge multinucleated cell. By the way, why do they keep the nuclei? I mean, you can imagine you got to fuse them together, cells together, to get enough cytoplasm. But why don't they just make do with one nucleus? Yeah, Justin.
You make much more protein.
You've got a lot more machinery to make proteins and the like, right? Also, transcription and all that stuff, transcription and translation. It's not, you don't want to have to wait for the transcription translation to happen here and then wait for those proteins to passively go to the other end of the muscle fiber. Remember, you've got some muscle fibers in your arm, right, that are 10 inches long. Well, don't worry. It's full of nuclei and they're all busy active. And what we're going to see later is the nuclei at the neuromuscular junction. They're making some proteins that the other nuclei aren't because they're not needed. Okay. So again, if you're going to make a huge cell like a Purkinje cell, right, has 2 nuclei, or muscle cells, you're going to need more nuclei. As the myotubes mature, they start making the contractile machinery, myosin and actin, stuff like that. And that's when, for voluntary muscles, they start getting this striated appearance, where you look at them and you see these stripes that are running perpendicular to the length of the muscle fiber. Right. And that's why they're called striate muscle fibers. And that's the characteristic of muscles you control voluntarily, as opposed to smooth muscle fibers and glands, which don't have that appearance. And at that point, we call them myofibers, or myofibrils, or stuff like that. And eventually, the myofiber, the muscle itself, comes together. How is that? So, in order for a neuromuscular junction to form, there is basically a dance with 3 partners. There is the presynaptic axon terminal that came from a motor neuron. Let's say it's a motor neuron in the spinal cord, sent its axon out the ventral root, all the way out to the body, one of the longest axons you've got in your body, and eventually contact a muscle fiber. And there's the target muscle, a myofiber. And then another important factor are Schwann cells. So not all peripheral axons are myelinated, and not all myelinated that much. And what is it that axons do in the periphery? What do they provide to tell Schwann cells to start? Oh, I don't have it there. Anyway, we'll run into it again. So the axons are telling Schwann cells to put together. And what I was going to say is, not every peripheral axon is myelinated, but every neuromuscular junction has a glial cell associated with it a Schwann cell, and they're often called a terminal Schwann cell. And these Schwann cells engulf the neuromuscular junction and basically keep other things from getting in there. So they're just putting a barrier around it to keep acetylcholine from getting out and to keep other factors from getting in. Not foolproof, but that's one of the jobs it's doing. So here's what the mature neuromuscular junction is going to look like. Where in the muscle fiber, there are these deep troughs that are characteristic of neuromuscular junctions. And the acetylcholine receptors are up here at the peak of these troughs because that's where they're going to run into the acetylcholine first. And the various troughs that are in between, the deep junctional folds, they are rich in acetylcholinesterase, which is going to chew up the acetylcholine. So you can think of these troughs as being places that get rid of any acetylcholine that has already done its job. Maybe the acetylcholine molecule bound to the receptor. As you know, it's a temporary binding process when things are working well. Then it unbinds, and if it gets into these troughs, it gets chewed up. And the various components of acetylcholine can then, to some extent, get recycled to make more acetylcholine. And what else do I want to tell you about this? The brown stuff here is meant to remind you that the synapse isn't empty, right? It's full of these extracellular matrix proteins, enough that we'll even call it a basal lamina. Both cells secrete these things, fibronectin, laminin, stuff like that. And they are long chain molecules that like to bind each other more or less indiscriminately. And so you end up with, again, I keep using that word gamish. What else? Here, for Abder's sake, there are the calcium channels where the action potential, when it comes down, calcium is going to open up and trigger the acetylcholine release. And another characteristic of neuromuscular junctions, perhaps because they are so crucial, and it's surely related to the fact that they're so huge, is every action potential that reaches the end is going to cause the postsynaptic cell to respond. So you can stimulate that nerve 1,000 times a second, and the muscle is going to get the signal 1,000 times per second. The contractile machinery might not catch up, right? But you can do things where you put something in the bath, say you've got a muscle in a bath and the nerve leading to it. You can put something in a bath to keep the contractile machinery from working. And then you can put an electrode in the postsynaptic cell, and you can see that there's an excitatory postsynaptic potential that's above threshold every time. This is so-called safety factor. You don't want neuromuscular junctions to run out of acetylcholine and to stop work. Nevertheless, sometimes they do, right? In emergency situations, when things are not going so well, some of the reason that the muscle doesn't respond is because the contractile machine, is because the neuromuscular junction isn't quite keeping up. It's running low on supplies. There. The creation of this synapse is a matter of mutual induction between all three players. So, here's what we know, is that when the motor neuron growth cone first contacts the muscle, as I've told you before, the motor neuron growth cone is already releasing a little bit of acetylcholine, more or less all the time. And the muscle fiber has already started making acetylcholine receptors and inserting them in the membrane. What's interesting is before innervation takes place, the muscle fiber is making acetylcholine receptors and inserting them over the entire muscle fiber. And that's probably to help the process so that wherever the motor neuron axon growth cone first contacts the muscle, there will be receptors there to respond to. But as the neuromuscular junction begins developing, the acetylcholine receptors start clustering there. And at first, all the nuclei in the muscle fiber are making acetylcholine receptor proteins and inserting them in the membrane. And in the initial stages, those membranes, remember I keep telling you these transmembrane proteins are floating, those acetylcholine receptors that were made beforehand physically move toward the junction, toward where the axon touched. And eventually, it'll be the nuclei here at the so-called junctional nuclei that will be just about the only ones that are transcribing the acetylcholine receptor subcomponents. And that's where almost all of the acetylcholine receptors will get inserted, right there where they're needed. Questions about that? And we'll talk the details about that in a moment. So one of the signals passing between the presynaptic and postsynaptic cell is indeed the neurotransmitter, at least in the case of the neuromuscular junction. And then they both start contributing to the extracellular matrix there. And as it points out here, one of the things that's especially rich in the extracellular matrix at neuromuscular junctions is stuff called beta-laminin. So, in fact, it's a synapse-specific laminin protein. And so, Josh Sains, who was the one who first figured out that there was a synapse-specific laminin, he dearly wanted to call it sclaminin, but that was seen as too undignified, and so he gave in and said, okay, well, it's also the second beta-laminin family, so it's beta-2 laminin. It would have been so much easier for you to remember if they had left it as slaminin, but there you are.
Hey.
So another thing about neuromuscular junctions that will be more of a factor in coming chapter 8 is the fact that at first, almost every neuromuscular junction will be innervated by more than one motor neuron. And what happens as development proceeds is at every neuromuscular junction, one of the axons retracts. And you can actually see the result of it, the so-called retraction bulb, as it lets go and gives up. And we'll see more about how this happens later, but I want to point out to you that in the adult muscle, 99.9% of the neuromuscular junctions will have only one input. Whereas in the newborn muscle, 90% of the muscle fibers have several inputs. And when the retraction takes place, you never end up with a muscle fiber that still has two inputs, and you never end up with a muscle fiber that has no input. And so think about that, and you'll realize that the only way for that to happen, for the number of inputs per muscle fiber to get smaller but never get to zero, is there's got to be two-way communication between the two, right? Because otherwise, if it was just a matter of the presynaptic cells withdrawing lots of axons, you'd think there'd be some muscle fibers that would get orphaned, but it doesn't happen. More about that later. Questions? Oh, and this is what we're showing here. Multi-innervation early in life, single innervation in maturity. Oh, and the other thing that happens is once one motor neuron input remains behind, that's when the junction grows enormously. So that you have this really huge synapse, a synapse so big that you can see it with your naked eye. Because you really don't want that connection to fail, right? You need the muscle fiber to contract when you need it to contract. Otherwise, you're not going to leave any baby behind. Okay, so here are some of the inductive factors that are going on. Motor neuron axons release a secreted protein called Agrin, and it got that name because it induces the muscle cell to start aggregating acetylcholine receptors at that site. So, just to complicate your lives, Muscles make Agrin too, but they make different isoforms. And for everything we're going to be talking about, the motor-on version of Agrin is 1,000-fold more effective than the muscle version of Agrin. So from this point on, we're going to forget that muscles make Agrin because it's not relevant. Rather, it's the motorons that are releasing their version of Agrin, and it is detected by muscle fibers by a complicated receptor with two components. One part of the component of the receptor is the muscle-specific kinase. So it's a protein that's only made in muscles, and it binds together two other proteins that we're just going to call LRP4, and they're the ones that are actually going to respond to the Agrin. So the receptor for Agrin is two components, right? MuSK and LRP. When the Agrin binds to the muscle receptor, it phosphorylates the internal portions, which then causes the production of a protein called Rapsyn. Yeah, the same lab that wanted to call slaminin, they named this stuff Rapsyn. And Rapsyn's job is to bind the intracellular portion of acetylcholine receptors and lash them together. So you can think of the acetylcholine receptors at a neuromuscular junction as a flotilla of acetylcholine receptors that are all close together because their intracellular domains are being bound together, tied together by this stuff Rapsyn. And as I pointed out before, pre-existing acetylcholine receptors are also moving and are being attracted to this area and will eventually get lashed and added to the flotilla. So you'll end up with millions of acetylcholine receptors at every neuromuscular junction. So, yeah.
Or acetylcholine.
How are they moving? What's pulling them there? I don't know, and I'm not going to talk about it. But they can move, they can float around. Some of it's passive, but it's pretty clear that they're actually being recruited. In the early days, it would be, they were looking in culture, and they would use a bungro toxin, which is a toxin that snakes use, and they kill you because they block your neuromuscular junction. They bind irreversibly to acetylcholine receptors, but it was cool that you could add a rhodamine molecule to the bungro toxin before you put it in the bath. And so they would see a muscle fiber that's not innervated, and they see the rhodamine is fluorescent the entire length of the muscle fiber, right? Then they stop putting any more rhodamine in the bath. Now the motoron touches the muscle fiber, and all that fluorescence that was in other parts of the muscle, it starts moving. And in real time, you can watch all the fluorescent acetylcholine receptors get aggregated there where that motoron touched it. And one of the things the motoron touching did was to release
How did you say that Rapsyn was made?
How is it made? It is induced by Agrin. So Agrin binds the receptor and induces the postsynaptic cell to make Rapsyn, and basically it phosphorylates Rapsyn, and that's what makes it stick to the insides of the acetylcholine receptor. We still haven't made a neuromuscular junction yet, but here's among the other factors that are going to be important for this to take place. The neurons are also making neuregulins, and it's the neuregulins, you remember, that tell Schwann cells in the neighborhood, bind me. And if there's lots of neuregulin, there'll be lots of Schwann cell binding and a thick myelin sheath. We didn't talk about it before, but the neuregulins bind to something called an ERB receptor, another RTK protein that we saw way back then. And yeah. So one of the things that binding the ERB receptor does to the muscle is boost the production of acetylcholine receptor subunits. So remember, every acetylcholine receptor consists of several different subunits, alpha, beta, etc., each of which is encoded by a different gene. And these proteins bind together to form the canal that will have in the middle of it the ion channel that will eventually let stuff into the cell. And this neuroregulin, because the signal's going to the part of the muscle where the neuromuscular junction is, that's one of the reasons why there'll be enhanced transcription of acetylcholine receptors right there at the neuromuscular junction where you need them to go. And so, so we call those nuclei there either terminal nuclei or subsynaptic nuclei. So one of the reasons there's an enrichment for acetylcholine receptor at the neuromuscular junction is because of that process. What's the process? Neuroregulin binding to ERB-B in the muscle fiber, right? And ERB-B then boosting transcript. Okay, so before the neuron gets there, the muscle's making receptors its entire length. Once the neuron gets there, it starts making the receptor only where the contact took place. By the way, all your neuromuscular junctions are more or less in the middle of your muscles, which is interesting because when the muscle first got made and first got innervated, the muscle was a lot smaller than it is today. And probably the reason it's in the middle is because that means the growth bone got to about the right place. And they seem to be aiming for the middle of the muscle. Probably tells you there's some signal coming from the muscle and the growth cone's zeroing in on the height of the signal. What's interesting is that if you denervate a muscle, and by that I mean you cut the motor nerve that normally innervates it, once that's cut, of course, you no longer have control of that muscle. But unlike the situation in the central nervous system, in the peripheral nervous system, you are capable of regenerating. So if you stay alive and rest long enough, the motor axons will make new growth cones and find their way back to the muscle and reestablish innervation, and now you get control of your muscle again. This is why you really don't want to be cutting peripheral nerves, right? Because you denervate muscle. What's interesting is that when that happens, there's a repeat of the ontogeny of the whole system, because muscles, once they lose innervation, they start making acetylcholine receptors all over their bodies again, the entire length. Everyone get that? In part because they're no longer getting that Agrin signal to tell them, and the neuroregulin signal to tell them to make acetylcholine receptors here. They start making it everywhere. And that's probably good in helping to reestablish contact once the motor growth cone finds its way back again. Another thing that happens, though, is that, and I just haven't said, so the motor neuron releases a whole bunch of acetylcholine into the neuromuscular junction. The muscle fiber has those acetylcholine receptors that cause it to depolarize. The depolarization of muscle fiber looks just like the depolarization of a neuron. And muscle fibers also make action potential. So the action potential begins at the neuromuscular junction and then spreads both ends of the muscle fibers, and eventually allows ions to enter throughout the muscle fiber, and they trigger the actin-myosin system so that the whole thing contracts. Got that? So, yeah. Yeah, Maria?
Neuroregulin is also used to help Schwann cells demyelinate.
What's also used to help Schwann cells?
Neuroregulin is also
Neuroregulin, yes, yeah.
Schwann cells. I believe the system to myelinate the axon.
Yep.
In the PNS. Then how does neuroregulin know exactly if it's helping to assist with the transcription of these acetylcholine receptors, or if it's supposed to help with that?
It's a great question. No, the neuroregulin, and of course the neuroregulin molecule itself doesn't know anything, but the axon's making it. And some neurons make more neuroregulin than others, right? Because that's how they'll end up with more myelination than others. But in both cases, both the Schwann cells and the muscle fiber are using the same receptor to respond to the neuregulin, but they give different responses because they're different cells, right? Muscles are different cells than Schwann cells. And so, again, it's doing double duty. So, and you can measure the action potentials from muscle fibers easy, right? What do we call that? That measurement. Electromyogram, EMGs. That's what they're doing. They're measuring the huge action potentials that are going up and down the muscle fibers, where you can have electrodes on the outside of the skin and you'll detect just fine. With deinnervation, well, that depolarization of the muscle fiber that goes up and down, another reason the acetylcholine receptors are concentrated in the neuromuscular junction is because it's the nature of muscle fibers that when they are depolarized, the acetylcholine receptors in that area tend to be destabilized and fall apart. And so why do they stay at the neuromuscular junction? Because there are these inductive factors that keep telling the muscle fiber, keep making acetylcholine receptors here. So in general, muscle depolarization is bad for acetylcholine receptors. And that's why the acetylcholine receptors that do get inserted the length of the muscle, if they're away from the neuromuscular junction, they're inhibited by that depolarization. Everyone, is your cognitive load high enough yet? Right? It's a complicated system, Justin.
I didn't understand that.
Okay. It is the nature of acetylcholine receptors in a muscle fiber that if a depolarization happens there, that tends to cause them to come apart, to disassemble.
Okay.
And so, well, wait a minute. There's depolarization in the neuromuscular junction too, isn't there? Yes, although no greater than anywhere else in the muscle fiber, right? Once the voltage-gated sodium channels open up, they're going to go the entire length of the... So, the depolarization is just as high at the end of the muscle fiber as it is at the neuromuscular junction. And depolarization causes the acetylcholine receptors to become unstable. But at the neuromuscular junction, there's so much inductive call for, let's have plenty of acetylcholine receptors, that they stay. Also, the Rapsyn binding together also tends to stabilize the acetylcholine receptors at the neuromuscular junction, and that's not happening outside the neuromuscular junction.
Kind of a way, like beyond just bringing them over the receptor, is also a way to get rid of other ones that shouldn't eat.
Right. If you really belonged here, you'd have Rapsyn on the inside. Get lost. You're not serving any purpose here. Yeah, Amy?
I like things at back here on the junction are mostly located in like the middle of the muscle.
Yeah, I shouldn't. Yes, it's true. It's not really relevant to anything I'm saying today, but I couldn't resist. Yes.
But like, when it's not like innervated, you said that like the receptors are like all over it. Is there like a reason?
True. Is there a reason?
Like, doesn't that it seems like maybe a little wasteful?
It absolutely does, right? So on the one hand, but on the other hand, there's not that much acetylcholine receptors getting made because there's no longer the Agrin induction saying, let's go with the acetylcholine receptor. So it still getting made, it gets inserted everywhere, and there's not much depolarization of muscle taking place because it's never getting that acetylcholine dump that says, hey, contract. And so, so it reaches some level of stasis there. Yeah.
Is using too much of something like succinylcholine depolarize and stay there and cause paralysis? Is that associated with weird aggregations of acetylcholine receptors?
But you're saying there's too much acetylcholine.
If there's too much succinylcholine, there's going to be too much depolarization.
And the muscle will keep contracting and contracting and contracting. Yes. Yeah.
You'll have too much depolarization, so that would destabilize the acetylcholine receptor.
Oh,
weird aggregation if that's happening.
No. And does that then lead to abnormal aggregation?
Those who
Is it not that I know of? No. So it's all a balancing act of what inductive factors are you getting and how much depolarization is happening. So yeah, and as it says here, with deinnervation, there aren't as many acetylcholine receptors, but they're distributed everywhere. There aren't as many and so it's not as wasteful, perhaps, Amy, as you might think, but there aren't as many because those inductive factors aren't getting there. So here then, remember, trying to teach is to tell you the truth as slowly as possible. Here's the figure to try to put all of this together. Okay? So the motor neuron is releasing Agrin and Neuromuscular, right? So the Agrin is causing a postsynaptic cell to start lashing its acetylcholine receptors together there. And the two are, doing lots of stuff. It's also releasing neuregulin that's telling the terminal Schwann cell to start becoming not just any old Schwann cell, but a terminal Schwann cell. We're making a synapse here, and so you've got to make the umbrella to keep the rest of the world out. Just to make things harder for you, Schwann cells not only respond to neuregulin, but they also release neuregulin. And they too are urging the postsynaptic cell to start making plenty of synapse, right? Start making all the things that are needed to make a big neuromuscular junction. Those extracellular matrix proteins are also getting made by both sides and helping keep everything together, etc. And outside the neuromuscular junction, electrical activity tends to reduce acetylcholine production, clustering, and half-life. So a dance for three, right? All three of these things are coming together and doing their job to make the neuromuscular junction exceedingly effective and to keep it present and to keep everybody else out. And this is the simple story of how synapses happen. And we know the most about it because it's a great big synapse that's out there in the periphery where it's easy to study. Presumably in the central nervous system, there are going to be at least as many players and at least as many signals going back and forth to decide whether the synapse will stay, go, get stronger, get weaker, stuff like that. This is the simplified view. And so that we can start with a Kahoot next time, I'm going to show you one more slide. This is a dense figure, right? So take it slowly. Go back and look at the simpler figure first and work your way through that, and then come back here and see that. I haven't even talked about the fact that the muscles is in fact sending signals to the presynaptic cell too. A difference, I don't even show neural ligand there, but are doing that, all of which is causing the presynaptic cell to start making more presynaptic machinery, stuff like that. Last story to tell has to do with extracellular matrix and basal lamina. In some pretty cool experiments, scientists killed in frogs, they killed the muscles by taking big slices out of them on either side of the neuromuscular junction, and then they cut the motor nerve. And then they waited for the motor nerve to find its way back in. And what they found was that even before the muscle fiber had recovered, it's dead. All that's left there is the basal lamina ghost, the sheath that was there before, and yet still the motor neuron axon would first contact the place where the synapse used to be. And so that shows that the molecules in the extracellular matrix serve a function, and that the motor neuron growth cone can recognize that place and will preferentially insert there, preferentially innervate there, and speed along the reinnervation and therefore the recovery of function of the muscle. Okay, so save up questions. We'll do them next time, and then we'll start with a Kahoot about all this stuff. I thought I'd get to 2 Kahoots today, but it wasn't meant to be.
I was pretty